a lot of copies of a little protein or not very many copies of a big protein? Which has more? So if we think about it in terms of mgs per mil, well, then the big protein might win. But in terms of actual copies, then the little guy is the winner. And so when it comes to protein concentrations, we don't just need to know the mgs per mil, which is what we can typically measure with something like a Bradford or UV, but we need to actually know like the copy numbers. And this is typically going to be done in terms of molarity or like micromolar and things like that. But that's going to deal with copy numbers. And in order to get between the copy numbers and the concentration in terms that we can measure, we're going to have to do some calculations. And so today I just wanted to go over how these calculations work, as well as some places that they come into play in the lab and why we really should be thinking about these sorts of things more than we are. I'll get much more into this, but each protein has a molecular weight that's going to allow us to convert between the mass of that protein and the number of copies of that protein. These molecular weights are often given in Daltons or kiloDaltons. Sounds really scary, but a Dalton is just a gram per mole. And then a kiloDalton is 1,000 grams per mole. And so we'll get more into these concepts, but you might be familiar with molecular weights just from like molecular weight standards when you run a gel. And just a little PSA before I get too far into this, that the bigger proteins are going to show up deceptively strong on these gels because they have more mgs wise in the same amount of copy wise as a small protein. And so a small protein, if you have a faint band, don't freak out just yet. Big protein, you got a strong band, well, still good, but it might not be as good as you think if you think about copies. So why should you think about copies?
Care about the number of copies for a number of different reasons, and sometimes we care more about it than others, and sometimes we should care more about it than we actually do. It's kind of like if you had a pound of feathers and a pound of bricks. They're both going to weigh the same, they'll both weigh a pound, but there's going to be a lot more feathers than there are bricks. And so we can easily get deceived by having equal like amounts in terms of masses or equal concentrations in terms of mg per mil, but having drastically different amounts of proteins. So we would need a lot of copies of a small protein in order to have the same actual number of copies of a big protein. And this can have big effects when we're talking about how molecular interactions are actually happening. Because these molecules, you're typically dealing with molecules interacting with one another in specific ratios. And we can talk about these ratios as like stoichiometry. So you might have like a 1-to-1 stoichiometry where you have one copy of protein A, say, and one copy of protein B, or you have 2-to-1, you have 2 of A and 1 of B, or 1-to-2 and 1 of A, 2 to B. You can have multiple, even more. So we can have these different combinations, but all of this is dealing in terms of copies. And if we have mg per mil, that's not going to tell us about copies. In order to get that information, we're going to need to know the how to convert from copies to the actual, or from mgs per mil or whatever the mass we're given to the actual copy number. When we're talking about copies of things, we're typically dealing with molarity and moles. So a mole is 6.02 times 10 to the 23rd, which is Avogadro's number. And 1 molar means 1 mole per liter. So it's kind of like saying that there are a dozen bagels in a box, except here we have a lot bigger number because these molecules are really, really tiny, and we can therefore stuff a lot of them into a small space. Even though we're stuffing a lot of them in there, we're typically not dealing at the same levels as if we were, say, making a salt solution or sugar solution. In those cases, we might be dealing with millimoles, so like thousandths of a mole, or molar even. With proteins, we're often dealing with micromolar, nanomolar, picomolar. So it helps to have a good sense of being able to convert between these using metric prefixes, noting that a micro means millionth, nano means billionth, pico means trillionth, and in between these, you can convert between them by multiplying or dividing by 1,000. And to get 2 levels up, you do by 1,000,000, et cetera, et cetera, et cetera, and more in metric conversions in another post. But you should recognize that you're probably going to be working in the micro nano sort of range when you're dealing with proteins concentrations. But in order to even know that, you have to be able to convert from the mgs per mil that you were measured or that you were given. I've been talking in the last few posts about different ways that we could talk about molecular weights and converting between units. And then we get to proteins, and we're in this thing called Daltons or kilodaltons. So when we talk about molecular weights, that's how much one mole worth weighs. And when we talk about Daltons or kilodaltons, well, a Dalton is how much a mole weighs, but it's already takes into account the grams per mole. So typically when we think about molecular weight, we think about it as being in grams per mole. And Dalton, you can think of it as kind of like shorthand for meaning grams per mole. So 1 Dalton is going to be 1 gram per mole. Often these proteins or other macromolecular complexes, things like large DNA or RNA, or maybe some complex of RNA and DNA, they're going to be really massive. And since they're so big, we're going to actually need to go up in our metric prefixes and go to kilodaltons. So a kilodalton is 1,000 Daltons. So remember, Dalton is a gram per mole. So 1,000 Daltons is going to be 1,000 grams per mole. 1,000 grams is the same as saying 9 kilograms. This is the same as a kilogram per mole. So if you want to go from Daltons to kilodaltons, you're going to divide by 1,000. So you should be getting a smaller number because it's kind of like you're thinking. You have like a car full of pizza carrier bags full of pizza boxes full of pizzas, and each of those pizzas have slices. If you're thinking in terms of how many slices you have, that'd be like thinking in terms of Daltons, and you're going to have a lot of slices, but you're going to have fewer boxes. And so would be like your kilodaltons. So to go from Daltons to kilodaltons, you're going to divide by 1,000, move your decimal point to the left 3 places. And to go from kilodalton to Dalton, you're going to multiply by 1,000 because now you're thinking in terms of pizza slices. And so you have to multiply by how many pizza slices you have per pizza box. Things get kind of confusing though when you're thinking about these various conversions where you're actually dividing by the molecular weight or multiplying by 1 over the molecular weight, which is the same as dividing by the molecular weight. But it can help you see how the units cancel out. So in all of these cases, it's good to have an intuitive sense of whether things should be getting bigger or smaller. So keep going back to your idea of those pizzas and the pizza boxes and thinking about do things make sense. Also line up your units in dimensional analysis, make sure those units are canceling out. There's this kind of like back of the hand estimate that the average amino acid is about 0.1 kilodaltons, so 100 Daltons, because remember that to go from kilodaltons to Daltons, you multiply by 1,000, moves that decimal point 3 places to the right. So the average amino acid is about 100 Daltons. So if you had a 1,000 amino acid long protein, you would have a molecular weight of about 100 kilodaltons. Well, remember that we could write this as 100,000 Daltons. But you can see here that 100 is a lot nicer to work with than 100,000, and so kilodaltons often come in handy. We can also go the other direction. So if we had a protein that was, say, a certain number of kilodaltons, and then we can multiply that by 10 to get the estimated protein length. So if we started by knowing that our protein was about 100 kilodaltons, or then we can multiply it by 10 to figure out it was about 1,000 amino acids long. And if we wanted to know the estimated molecular weight from the protein length, we could go from the number of amino acids to divide it by 10. So if we knew that it was 1,000 amino acids long, we would divide it by 10 to get to 100. Okay, so these are just estimates though. And the actual amount is going to vary from protein to protein because proteins are made up of different combinations of amino acids, and these amino acids vary greatly in their sizes, from this giant old tryptophan to the teeny tiny glycine. And so since proteins are different combinations of these, in addition to having different numbers of these, like different lengths of these, the actual conversion isn't going to just be this nice little estimate. And if we really care about the conversion, then we're going to need to know the molecular weight, the like exact molecular weight and stuff, which we can figure out by looking it up online with tools such as XBasy-Prop-Param.
So we often think about the molecular weight when we're thinking about like where our protein is going to run on a gel. The longer proteins are going to get tangled up more in that gel mesh and they're going to be higher up. but the size is also going to influence how strong that band is. So we typically think about like the banding when we're just like putting a dye to see where the protein is in the gel. The stronger the band, the more in terms of like mgs per mil typically, but not necessarily in terms of copy number. So if you had for the bigger proteins, remember they're going to have like fewer copy numbers is going to give you the same amount of mgs per mil. So you could have a strong band for a bigger protein and a weak band for the little protein, and the little protein could still have more. So if you see a big band but your protein's pretty small, don't be too worried yet. The weight also comes into play when we're dealing with spin concentrators. And so these allow us to concentrate proteins, also like dialysis membranes. All these numbers like 10K, 50K, 100K, that's referring to kiloDaltons in terms of the molecular weight cutoff, or this like MWCO. I don't know where it is. I guess they don't have it written on here. But there we go. There's one with MWCO. So that's telling you like how much, like the size of the pores in those membranes. So you want to use a membrane that's going to keep your protein in the not let your protein through. So keep it in the little bucket part, and then everything else goes through. Because it's an average size, you want to use a membrane that has a cutoff that's about at least a half, preferentially like a third of what your protein actually is.
Once we have that exact molecular weight, we can do a lot of things with it based on based on what we know. So often, again, what we know is going to be the mass to volume. So you might be able to calculate this using something like a Bradford assay or based on its UV absorption. These are going to give you mgs per mil. So even if you're using a UV absorption method where you measure basically the concentration based on knowing that a certain amount of a certain protein will absorb a certain amount of light, and then measuring how much of that light it actually absorbs, and then going and converting that to how much of that certain protein was actually there. So this is like specific for that protein, but it's still going to give you the answer in mgs per mil. And so then you would need to convert it back into your molarity, if that's what you're caring about. So you can get to molarity by dividing mass volume by molecular weight. And remember that, so when you're dealing with molarity, that's moles per liter. And so if you're in moles per liter, and when you're dealing with the molecular weight, typically that's grams per mole. If you're in kiloDaltons, then you're in kilograms per mole. So you're going to need to convert to Daltons in order to use this equation as written. And so remember that you can do that by just, in this case, you would divide this whole thing by 1,000, or you would multiply your molecular weight by 1,000. But then since you're dividing by it, it's like you're dividing this whole thing by 1,000. But there's a shortcut. So say, so often, in addition, like the mass per volume, this is often mgs per mil. And you might say, okay, well, now I've got to convert my mgs to grams. I've got to convert my mils to liters. Well, think about how you would do that. You would multiply them both by 1,000. So you'd be multiplying by 1,000 over 1,000. And that's the same as multiplying by 1. So, and like if you were to line it up, then the thousands would cancel out. So it would be, you would just be like undoing what you just did, but you'd be getting the same number. So basically, if you're dividing it by the same factor, then you can it's just the same number. And so when you have mgs per mil, that's the same as grams per liter, and it's also the same as micrograms per microliter, which can come in handy when you're dealing in the microliter scale and want to know how much protein is in what you just pipetted out. So you can use this shortcut and therefore use mgs per mil instead of grams per liter because it's the same thing. Okay, so molarity, you can take your mgs per mil divided by your molecular weight in Daltons, or if you're in terms of kiloDaltons, you can either explicitly convert to kiloDaltons beforehand, or just do it when you're convert to kiloDaltons beforehand, or just do it directly when you're doing this measurement. You can also go the other way, get your mass to volume from your molarity and your molecular weight. And you can get the mass from the molarity and the volume. If you want to know how much you have in your tube, maybe you're concentrating your protein down, and you want to know like how much to suspend it in or various things like that. It can be helpful to know the actual mass that you have. Or maybe you have this, like different versions of the same protein, and one copy of like maybe the protein, someone expressed it with a tag on the end. So basically we can stick these extra sequences on the end of proteins when we're expressing them recombinantly. So when we take the instructions for making a protein and we stick them into cells, like bacterial cells, and tell the cells to make it for us, since we're putting the instructions in there, we can add a little bit extra. And this is going to make a little extra few protein amino acids on the end of the protein that are going to help us with like purification and stuff. Sometimes we cut these tags off, sometimes we don't. And so you could have the same like protein in terms of like you could have the same stoichiometry with that protein and other proteins, but one of the proteins would be bigger because it had that tag on it. And so now you're going to have a different molecular weight of that protein. And we need to be able to take that into account so that if a company is, say, selling a protein that has that's a tagged version, and they say, oh, we've got 20 mg per mil. Well, is that really a better deal than like a protein that was 20 mg per mil, or 10 mg per mil, but didn't have a tag? Well, it's going to depend on the molecular weight of the tagged versus the untagged. And so the molecular weight this conversion is going to come into play there too. So let's look at an actual example. So say you have a 30 kilodalton protein, we'll call it protein A, and we have a certain concentration of 5 mg per mil. And then we have a 90 kilodalton protein. So this is our protein B, and it's at a concentration of 10 mg per mil. So first, without doing any like formal calculations, just thinking it through, which of these solutions has a higher molar concentration? So which is going to have more protein copies per mil? Well, protein B is going to be 3 times more massive than protein A, because we said it was 90 kilodaltons, protein A was 30 kilodaltons, 90 divided by 30 is 3. So it's going to take 3 times as much mass of protein B to equal the mass of protein A. So what about, we know the concentration of our solution in terms of mg per mil. Our protein B solution only has 2 times as much mass-wise, so protein A is going to have a higher molar concentration. So now let's actually show that math-wise. So remember that we're dealing with we're giving kilodaltons, but we need to use Daltons. And so we need to work with grams per mole instead of kilograms per mole. So we can do this all with dimensional analysis, doing it all formal, canceling out all of the units, or we can take some shortcuts. So for example, if we recognize that 5 mg per mil is equal to 5 grams per liter, because remember, you're multiplying both of these by 1,000, you can see here, 1,000, those cancel out. And so we can say we can just start with 5 grams per liter. Then we just have to multiply it by our mass divided by our volume, by our molecular weight. And then we get the answer of 1.67 times 10 to the minus 4th molar. This should be like screaming out to you, convert this, convert this. So if you're dealing with -4, the easiest way to like convert this between to a unit that avoids scientific notation, well, our options are going to be 10 to the minus 3rd, so a milli, or 10 to the minus 6th, a micro. And so if we're dealing with 10 to the minus 3rd, we still have a decimal, so let's deal with micro. And so if we multiply this by 10 to the 6th, well, now we can move that decimal place over 6 points. This is going to give us 160 micromolar. So we're going from molar to micromolar, and we're getting 167. And note that we're getting a bigger answer because we're talking in terms of the same amount of stuff, but in smaller units. So thinking about in terms of pizza slices rather than pizza boxes. We can do the same thing for our protein B solution, but here we just have to put in 90 kilograms per mole because we have a 90 kilodalton protein, and 10 mg per mil instead of 5 mg per mil. And now we get 1.1 times 10 to the 4th molar. So we get 111 micromolar. And so we can see that the concentration of protein A was higher, even though we had in terms of its molar concentration, even though we had a lower concentration in terms of mg per mil. And we can similarly convert between all sorts of different scales, and more on this in my post before. So it's helpful to know, be familiar with molarity, be familiar with these various prefixes and dimensional analysis, and how to convert between these different units, deal with scientific notation, and think logically and do like reasonableness checks when you're doing your calculations. Additionally, we don't just care about how much, how big, how many copies. We also care a lot of times in terms of the activity when we're dealing with enzymes. This is like how much the thing can actually do. So maybe some of those copies were defective. And so if those copies were defective, then they wouldn't be very helpful to us, and they would have, therefore, would have a lower specific activity, which is telling you about how much the different copies can do. So this is another reason, this is another way we can kind of control for differences in concentration, thinking in terms of the concentration and in terms of the activity, which we can do when we're dealing with enzymes. So when we're dealing with reaction speeder uppers, which might be something like a restriction enzyme, so something that cuts DNA in specific sequences, or a ligase, which can stitch together pieces of DNA, a DNA polymerase, which can stitch together DNA letters into chains, various things like this. So in that case, we might be wanting to deal with specific activity as our measure of a sort of proxy concentration. So like we're talking about there, like the concentration activity rather than the concentration in terms of like copy numbers. So always remember that mg per mil is only part of the story, because proteins don't like work in terms of mg per mil. They work in terms of how many copies there are, and also sometimes how functional those copies are. So we need to be able to convert between these, and the molecular weight is able to help us do this.